Yeah, thank you very much, Leila, for the introduction, and also good morning, everybody. So yes, I am scientific project manager at Europa Imaging, and in particular in the project AI4Life, I lead the work package on communication, outreach, and training. So today I will give you some introduction about the project, but before I wanted to also introduce a bit what Europa Imaging is. So Europa Imaging is the European research infrastructure for biological and biomedical imaging. It's publicly funded by 17 member states that you can see in the map there. And there are plenty of nodes that are imaging facilities all across Europe. The whole point is that um, all of these different facilities are offering different technologies. So whenever a user needs to use some particular microscope that is not available in their facility or nearby, they can travel to a different node and perform their experiment with the particular technology that they are interested in. Oh, that works. So the portfolio, it's quite broad. It goes from the atomic level to the human level. And of course, there are also image analysis services associated to each one of them. This is all provided by the different Euro imaging nodes. And we have also image data services in that. So we want to have the fair data coming from these microscopes, but it's quite challenging in general. So the size is usually quite big. You can see some examples there from the different ranges, from more human size to cellular level. And this can take several terabytes. It's also challenging because it has different channels. You can see different colors in the image. It can have sometimes time points, so it becomes 4D very easily. Sometimes there are different technologies that are also correlated into just one data set, and that's some extra layer of complexity that is added to it. And the big challenge as well is the data format. So there's no clear file formats. There are plenty of vendors that are providing their proprietary file formats for both the binary data and the metadata as well. However, there is a lot of hope now with OMSR, that's a next generation file format that was released some time ago. It's becoming the standard, but I think that there are high expectations that this will solve all the problems. It might be or might not. And it includes also the metadata. So it's not only the data that is compressed in there, but also the metadata. And it's represented in kind of a cube, like you see at the bottom of the slide, that provides high access to the different slides. So based on the metadata, one can select the cube that is of interest. That is very convenient for cloud storage and in general to access remote store datasets. You can see at the bottom right of the slide an example of a slice of an organism that is quite big, it's 8 terabytes, and one can actually go super deep into the subcellular level in a very quick way and have a look at this part of the image. It's also called pyramidal file format because one can go slowly into these different layers and go deep into the image. So I also wanted to mention something that it's going to start soon that might be interesting for you. It's a new project that it's going to be coordinated by Europa Imaging. It starts in January and it's called Founding Guide. And the idea here is to connect different repositories in different locations. And there are some in Europe and Japan and Australia. These are the main partners of the project. And there will be some work on ontologies and metadata there because these resources will have to become interoperable. That's the whole idea. So we will cover both sides. On one side, the microscopy image data, but also the preclinical image data. In the clinical field, the file format and the analysis is way more standardized. So many people use XNAT for this. On the microscopy side, it's a bit more complex. So we have the bioimage archive in Europe, the IVR as well, and SSBD. And all these resources will be talking to each other in a easy way. Okay, so now I will start describing a bit what AI4Life is. So Europa Imaging is coordinating this project. There are 10 partners all over Europe covering 8 different countries. Of these partners, 4 are research infrastructures from different fields. It's going to last 3 years. We just finished the first one and it's a total budget of 4 million euros. The idea of the project is to bridge the gap between two different communities. On one side, the life science community that is producing plenty of data, but doesn't necessarily have the expertise to analyze using deep learning methods. And on the other side, the computer science community that develop those methods usually don't have access to data. So bringing them together to try to make use of those methods in life sciences. And we offer different services here, and I will go a bit deeper into those later. One is the bioimage model zoo, a repository for AI models, and then there is also the bioimage archive that is storing the image data sets. We also offer some engagement for the project partners, but also beyond the project in the form of open calls and challenges. So the idea or the main objectives of the project are providing access to these AI methods in a fair way so that anyone can take those methods and reuse them, and they should be open and everyone can also contribute to them. Also by establishing standards on both sides. On one is the description of the models, but also the descriptions of the data sets and the annotated data sets. And then making this sharing and dissemination of these things quite straightforward. We provide support for users, but also for developers that want to contribute. I mentioned before already the open calls and challenges. So the open calls would be for life scientists to support them with their analysis. And on the other hand, we have challenges for developers to compete and improve a particular method. We organize also plenty of outreach activities and training that I will mention later on. Okay, so this is the structure of the project. It's not so relevant for you, I guess, but just very briefly, besides the project management and the outreach, that is the part that I'm taking care of, we have one work package per service, more or less. So the work package 2 would be on the computing and the AI models repository. Work package 3 is more on support to the different users. Work package 4, it's providing support to the developers, so more technical side. And work package 5 is on the storage, so that's more the bioimage archive side. And in work package 6, we engage with the audience with open calls challenges. These are the partners of the project. So EMBL is part of it. It's EMBL and Human Technopole are the scientific coordinators. Bioimaging is the coordinator of the project. And we have different research infrastructure like Embrace, Emphasis, OpenScreen, and Instruct, and many other institutes all across Europe. This is the project partners. So those are the part of the grant, but we also have community partners. And those are all these software tools that want to be interoperable and are able to produce models and consume models from other tools as well. So I'll start now with the bioimage model soup, which is this repository that I was talking about already. The idea here is to have AI models in a homogeneous way that can be also run by different tools. So you see here a screenshot of how it looks like. There are model cards for each one of the models, and the tools are on the top. So those are the community partners. This is growing constantly, and one can take a model, download it, and reuse it, and open it with the tool that one is more familiar with. It's totally community driven. Anyone can contribute to the code, and it's totally open source. And there are some guidelines also for contributing the models. Minimum rules are that the model should be able to run on at least one of the tools. It should be well documented. There are also details about this in the documentation. And they should be able to be reused by others. Then for the users, it's very easy because they would just go to the card, select the model that they're interested in, and download it. Yeah, so that's the bioimage model too, and the community partners, like I said, it's growing. There is a procedure for people to do this. In principle, they should comply with the general specification of how the model should look like, and that should be all for the different tool partners. Yeah, so the AI models are specified on a GitHub repository that you can see here. So this is the screenshot of the bioimage.io organization, but there are several repositories here that might be useful. So one is the specification, where one can have all the description of the models. So in principle, one have there the trained models. One needs to provide also an input image that would be an example of how to run the model, but also an example of what is expected afterwards. Because we display the cards, like I showed before, it's also good to have a cover image for representation of what is before and after applying that particular model. And then there's an important part that is the RDF specification. This is a YAML file that has to live in the same folder or zip file with all the fields that are mandatory there, with describing the architecture of the network, where the input and the output format, what happened before, how should be the preprocessing done, and also some performance metrics on this. All this is really described in this repository. I'm not giving so many details here, but you can take a look at this one and know a bit more. Then there is another repository that is also interesting. That's where there is a collection of the RDF model. So there is no proper API, but there is a file, there is a collection JSON file where you can find all the models that are at the moment part of the repository. There are also some scripts there that are useful tools, like to generate a particular model or validate them. This is something new that we are now working on. It's going to be released this week. This is BioEngine. So it provides the ability for users to run models in the browser. So they don't really need to download the model anymore. They could take their own images and run this. There will be a Kubernetes cluster on the backend running this with access to GPU resources. And they can really upload the images, run this for training and inference. We haven't published this yet, so it's at the moment, this week, this has to happen. So we are going to publish something on our website with all the details about this and how to test and everything. It provides really a multi-user platform and also multi-tools. Anyone can take it and take the model and run on the software that they want. So all of this is documented in the documentation. There's the bioimage.io/docs where one can find all this information. But also we are providing training and support. And we are now here collaborating with the Galaxy Training Network, where we deposit some tutorials there in plain text and then slides as well. And it automatically generates videos with captions. That seems to be quite useful for the community as well. If none of the above methods answer to user questions, then we have a help desk that will take care of particular questions or supporting people in, for example, creating a model or uploading the model or becoming a community partner. This is a preprint from yesterday. It's quite new as well. So they are now for support, they are developing this chatbot that it's being trained with the documentation on the bioimage model zoo, but also on these different community partners. And the bot helps you find a suitable method for your analysis. And I mean, I guess we are all familiar with ChatGPT, but similar format for image analysis. The preprint is now available, so there's a repository, it's linked there, and you can take a look at this one. We are also putting some effort on collaboration with industry. We are talking with different industry partners, but already with Leica, we materialize the collaboration. They are now taking the different models from the bioimage model zoo. They made it compatible with their software that it's called Avia. And now they display that in their application as well. So anyone can go through the particular commercial software and download the models and use them in Avia. So that was all on the AI model side. Now on the image data side, we also have another pillar important in the project, that is the bioimage archive. So bioimage archive stores image data in general, but with the project, it's also starting to include AI datasets, the image archive. So you see here an example. There's a gallery on the right side with all the different data sets that are available. They have been converted to OMXR. And on the left side, you see an example of an image. Maybe it's not very clear, but there's some kind of border or outline in yellow, red, and green. So this is the annotation of the image. It's not coming in the image naturally. So it just someone did that, either a person or a tool. And this is important for reusability of the data sets. The question now is, how can one store this in a fair way as well that other people can reuse? So for that, we had a workshop at the beginning of the year that was on these image annotations, because there are plenty of methods out there, plenty of papers that have been published with these results, but the image that are annotated are not available anywhere. So the idea here was to have these annotations in the bioimage archive, because it's usually quite resource consuming to produce them. And that can also help the method developers to take these annotations and improve their own methods or to train their methods. So that's why we think this is an important resource to be shared. The problem is, what is it that we want to store from these annotations? Yeah, that was the discussion that was happening in this workshop that we held together with the bioimage archive. So the conclusion of this one was to store metadata related to the study in general. So that's the same as for the regular data set. So keeping all this description and the licensing and so on, the versions and so on. Also describe what is the source image. The bioimage archive follows the REMPI recommendations. So what is it in there? What is the biological object that is being studied in there? And then there is a provenance as well. So who did the annotation and when did it happen? And how was the method that was used for this one? So was it the software? Was it the person doing that? How was it generated? Were many people working on this? Was it quality control? If there was a human annotator, how many years of experience? What is the confidence level of this particular annotation? And all these different aspects from the provenance. And then finally, more detail on the annotation. So what is it that you are annotating and where is this thing? Is there a bounding box? Is there a segmentation mask? What is it that is the region of interest that we are annotating in this particular image? Also, if the image was transformed. There were several days of very deep discussions about these topics. So at the end of it, and this is a preprint that will come out soon, the agreement was on reducing the number of formats, and like I mentioned before, next generation 5 formats and OMXR seem to be the way to go at the moment. There are also from other disciplines some other formats that can be also quite useful, and of course CSV and TSV for metadata. But for the images, OMXR seems to be the winner at the moment. Also, there's the need for standardized metadata, so having all these different annotations that I mentioned before, the confidence level, what was transformed, and so on. But also making all this data available for others to reuse. So that's for that an API is necessary, and that people should be able to browse and visualize these data sets before working with them. And finally, also acknowledging these contributions, right? Because the annotator should be credited properly there, and also especially when there are people that are contributing very heavily to these data sets, like data stewards, they're usually not rewarded, but that should be also something to take into account. Okay, so there are different opportunities to engage with AI4Life. On one side, we have workshops and hackathons that we are quite active organizing, and we have also plans for the upcoming year already. And open calls that I will describe a bit more. So basically, we are welcoming use cases from the different communities that have this need to use deep learning methods for their analysis. And a group of experts in the project will provide the support. So once they come up with a solution, the idea is that we launch a challenge when the data set is appropriate for that. And this challenge, in this challenge, different computational experts, we compete to improve the performance of the provided method. We organized plenty of these workshops and hackathons. So we started last year with the kickoff meeting in Heidelberg, but then there was some focus also in Java applications, and that happened also in Human Technopole in Italy. We had another one in Stockholm. We have another one this year again, like two weeks ago in Heidelberg. So there is a lot going on in this aspect, and we are going to organize much more in the next year. We have already 3 or 5 already planned. So that's something that we are welcoming everyone to join. So it's totally open. We open the registration, and if you are interested in any of these, you just need to register. Usually it has a topic. Sometimes people bring their own topics. And for example, when there is a tool that wants to become a community partner and they need particular support, they contribute, they join this event, and the community will help them. This year, when we had the second General Assembly 2 weeks ago, we organized a hackathon, but also in parallel, we had a solvathon, and there was different experts working on the open core project. So a bit more on this open call. So we launched this in February, and we asked the community if they needed some help with analyzing the image data or creating this training data, or in general applying deep learning to their research. We asked them many questions when they applied, so we kind of were at the same time running a landscape analysis that I will show a bit more later. But so we launched in February, and then the projects are running since July until the end of the year. Some of them have finished already, and some of them will be done by the end of the year. This is something that we done first time last year, but we need to repeat this 3 times all over the course of the project. And after each one of these open calls, we run one of the challenges as well. So we were surprised that we received so many applications of 72. So it's a significant number to analyze the landscape a bit. There was plenty of use cases from many different fields. Of course, cell is the most common one, but there are also plenty of cases on marine biology, and also plant and ecology. You see root, soil, of these things were quite often in the applicants as well. We asked them what is it that you need to do? What is the problem that you are having at the moment? And we offered them several of these options. They could select several. In general, everyone or most of them wanted to improve their image analysis workflow. And here we saw people that were not happy with the results that they had at the moment, but also people that were already happy with the results. And that was a bit surprising. So we asked having more, and they were happy with the outcome, but it was done in a completely manual way. So what they wanted from us is to automate this process. That's also some of the projects are about this as well. So that's the first case, but also the second most common thing was the example 4, so the consultancy on available tools and solutions. We interpret this as more need for trainings, and deep learning is everywhere, AI is everywhere, and people need to know more what is it that they can do with their data with this, and understand a bit how the field is evolving. Another question that we ask them is about the data that they have available, so the amount of data, file formats, all this, but also do you have annotations already? And most of them said no, or only a few examples. And then we ask them, for the ones that had some annotations, what kind of annotations you have? Silver would be the ones that are only machine generated, and then the gold one would be human created. So we see that we between the people that are they have annotations, there seems to be quite more on the gold level. This is anyway something that we are not taking. We are cautious in the interpretation, because it's in the end a self-assessment. So everyone can think that their annotations are gold, but maybe a silver. So it's not clear if this is totally how it looks here in the plot or not. So in the end, After the whole process, we had 8 open calls, 8 projects awarded. So they're on different aspects from skeletal muscle to marine biology. So there are some, sorry, on plankton and yeah. So there are all kind of different projects. Diversity spans across the different scientific disciplines, but also in countries, there are even SMEs involved in this. So everyone was welcome to apply, and we could see that diversity in the selection as well. All right, so with this, I think I more or less gave an overview on the project. We are meeting every Wednesday. Some of you join one of these calls. So we have, besides the regular meetings on the project, we have one for the bioimage model soon that it's running every Wednesday at 4 p.m. And everyone is welcome to join. You can just drop by there. We have all the events on our website. So as soon as they are confirmed, we publish them there. Not only the ones that we organize, but also the community partners are very active, participating in workshops, conferences, and so on. So all of this we try to reflect on the website as well. There is a newsletter that we release quarterly so that we promote all these different news and we make everyone aware of the different accomplishments of the project. And if you have anything that you would like to know more, you can just reach out to me and I can put you in touch with the person that is relevant for the particular topic. So thank you very much for your time, and I'm willing to answer your questions.